So now we're continuing our synaptic transmission. We have already defined what is the synapse. We have already defined what is a synapse. It is that gap between a neuron and another neuron, a terminal end of a neuron, and the dendrites. So the terminal ends of the axon and the beginning of the next neuron, these are the dendrites. So that is your synapse. So now when we examine synaptic transmission, we see that synapses are variable in their mode of action. So some neurotransmitters are excitatory. So they press the gas, while other neurotransmitters are inhibitory. And sometimes the transmitter's effect is going to differ depending on the type of the receptor on the postsynaptic cell. So some receptors are ligand-gated ion channels. So sometimes a neurotransmitter is going to bind an ion channel, while another one is going to bind a G-coupled protein receptor. So examples of the three kinds would be excitatory cholinergic synapses, where when you have acetylcholine that's gonna really leave the vesicle and bind postsynaptic acetylcholine receptors. There are also inhibitory GABAergic synapses. Inhibitory GABAergic synapses. These inhibitory GABAergic synapses are when GABA binds, that leads to down regulation, we will see. And excitatory adrenergic synapses, that is when your that's when your epinephrine, also known as adrenaline, that's going to cause the stimulation of a G-coupled protein receptor. So let's begin with an excitatory cholinergic synapse. So the cholinergic synapses have acetylcholine is their primary neurotransmitter. That's why they call them cholinergic. They're cholinergic because acetylcholine is their primary mode of action. So the steps in the synaptic transmission are the action potentials would depolarize the axon terminal, and that would lead to the opening of voltage-gated calcium channels. And then calcium would enter into the terminal end of the axon, and that would trigger the exocytosis of acetylcholine. Acetylcholine would diffuse across the synapse, binding to postsynaptic acetylcholine receptors. Upon the binding of the acetylcholine to the postsynaptic acetylcholine receptors are themselves ligand-gated ion channels that open and allow the influx of sodium and the efflux of potassium, initiating an action potential at the postsynaptic terminal. An entry of the sodium causes depolarizing postsynaptic potential, and if strong enough, that depolarization spreads to the trigger zone, and that raises the membrane potential up to the threshold to trigger an action potential. So that's the transmission of the cholinergic synapse. An action potential comes at the presynaptic neuron, leading to the influx of calcium. Calcium influx is going to cause the exocytosis of acetylcholine vesicles, and this acetylcholine is going to diffuse through the synaptic cleft, and it's going to bind to the ligand. So that's the ligand. Acetylcholine is the ligand binding to postsynaptic receptors, and the opening of the postsynaptic receptor is going to lead to the influx of sodium and efflux of potassium. That is now going to lead to step number 5, which is the action potential. Then the action potential is going to propagate downward, and that's how we mediate the action potential throughout the synapses. Acetylcholinesterase is very important. That's why some pesticides, if you go to a farm, they just spray it with pesticides. And sometimes in protest, they throw these gases. So what happens is these people, they have tear secretion, they're coughing because there's secretion of so much mucus. There is a secretion of nasal secretions and saliva. Why is that? Because these pesticides and these gases are going to inhibit ACHE. They're going to destroy acetylcholinesterase. So if you destroy acetylcholinesterase, that means you're going to have a lot of acetylcholine. And if you have a lot of acetylcholine, that's going to lead to overstimulation of postsynaptic terminal, and that's going to increase overcontraction of the gland, leading to so much mucus, so much tears. Okay, but normally acetylcholine is going to be destroyed by acetylcholinesterase. It ends with ACE. Acetylcholinesterase, that's an enzyme that will destroy acetylcholine, leading to choline being destroyed into acetate and choline. What they call it, acetylcholine. It's made up of acetate and choline. Acetate is going to be used by the body, while choline, what happens to choline? It's recycled right back. It's taken right back at the presynaptic terminal. An inhibitory GABAergic synapse. So the GABAergic synapse, known as the GABA aminobutyric acid. GABA aminobutyric acid is the neurotransmitter. The steps in the synaptic transmission of GABA are: you have an action potential. You have an action potential that triggers the release of GABA into the synaptic cleft, and GABA receptors are chloride channels. So once they bind to the postsynaptic cleft, chloride entry hyperpolarizes the postsynaptic membrane. So now we don't have excitation. We don't have depolarization like acetylcholine. Now we have hyperpolarization. And the postsynaptic neuron is going to be inhibited because it's not going through depolarization. It is going through repolarization. So no action potential, because GABA is a depolarizer with chloride. Now, an excitatory adrenergic synapse, that involves norepinephrine. But here, norepinephrine, we're not dealing with no ion channel. Here, we're dealing with norepinephrine, also known as noradrenaline. It is responsible for initiating a second messenger. Also, the monoamines and the neuropeptides, they bind to the G protein coupled receptors at the postsynaptic membrane. Therefore, they activate a second messenger known as cyclic AMP. And it is slower to respond than the cholinergic and the GABAergic synapses. They have the advantage of enzyme amplification. So a single molecule of norepinephrine produces vast numbers of product molecules in the cell. There we go. So now what do you see here? You don't see some vesicles that bind to postsynaptic membrane receptors. So here you don't see that a channel would open, like acetylcholine binding to acetylcholine receptor, leading to the opening of that channel, allowing for influx of sodium, efflux of potassium. And for the GABA, you have GABA that is secreted that binds to postsynaptic ion channels that allow for the influx of chloride, leading to hyperpolarization and inhibition. Here you don't see any of that. Here you see a G-coupled protein receptor. This G-coupled protein receptor would bind to the neurotransmitter norepinephrine, because norepinephrine cannot enter into the cell. And then the G protein is going to detach from the G protein coupled receptor, and then the G protein is going to cause adenyl cyclase. You see A's, cyclase, cholinesterase. So that's the second enzyme. But that's an enzyme on the plasma membrane that's going to cause ATP to be converted into cyclic AMP, cyclic adenine monophosphate. And that's going to cause multiple possible effects. So that's going to cause downstream enzyme target effects. That's going to lead to metabolic changes. That's going to may even cause genetic transcription binding to receptors that are inside the nucleus. leading to some enzyme synthesis that would terminally lead to metabolic changes, or maybe that's going to cause the transcription of ligand-gated channels that are going to be blocked in the plasma membrane, or lesion-gated channels that are going to open to cause depolarization. So to succeed the signal, it is important not only to stimulate the postsynaptic cell, but also to turn off the stimulus. The neurotransmitter stays bound to the receptor for 1 millisecond. So if the presynaptic cell continues to release neurotransmitter, one molecule is quickly replaced by another, and the postsynaptic cell continues to be depolarized, continues to be stimulated. To end the signal, the presynaptic cell stops releasing the neurotransmitter, and then the neurotransmitter in the synapse is going to be cleared by different ways, like the enzyme, like neurotransmitter degradation, like the enzymes in the synaptic cleft are going to break down the neurotransmitter, and acetylcholinesterase is going to break down acetylcholine into choline and acetate. Going to break it down into choline and acetate. And there's also reuptake, where the neurotransmitter or its breakdown products are going to be reabsorbed into the axon terminal. Example of that would be the choline from acetylcholine that's going to be recycled to make new acetylcholine. And you have amino acids and monoamines that are also going to be reabsorbed. And you're going to have degradation in the axon terminal by the enzyme monoamine oxidase. So sometimes we use for patients who are suffering from clinical depression and anxiety, we use the monoamine oxidase inhibitors. These monoamine oxidase inhibitors, they are very important because they are the targets, they prevent the reuptake of monoamine oxidase. So they prevent these monoamines from being degraded. So they propagate their action. They leave them alone. They leave them unbothered. So they keep stimulating postsynaptic monoamine receptors. And then we also inhibit and we cause the cessation of the signal by the diffusion. So the neurotransmitter or its breakdown products simply away from the synapse into nearby extracellular fluid. So there we go. The calcium enters, causes the vesicle to release acetylcholine. Acetylcholine will cause sodium influx, potassium efflux. And then acetylcholinesterase is going to degrade acetylcholine into acetate and choline. That's why we call it acetylcholine. Say acetylcholine because that's acetate. And we're going to break it into acetate. It's going to be used by the body, and choline is going to be reuptaken in the presynaptic terminal. There are neuromodulators. What are neuromodulators? Neuromodulators are chemicals that are secreted by the neurons that have long-term effects on groups of neurons. They may alter the rate of the neurotransmitter synthesis, and they may affect the release, the reuptake, or the breakdown. The release, the reuptake, or the breakdown of the neurotransmitter. And they may adjust the sensitivity of the postsynaptic membrane. They may adjust the sensitivity of the postsynaptic membrane. Nitric oxide is a simple neuromodulator. It is a gas that enters the postsynaptic cells and activates the second messenger pathways. For example, relaxing the smooth muscle cell, and neuropeptides are chains of amino acids that act as neuromodulators. You have enkephalins and endorphins. These are neuropeptides that inhibit pain signal in the central nervous system. When we discuss neural integration is the ability to process, store, and recall the information and use it to make decisions. So the chemical synapses allow for decision making. The brain cells are incredibly well connected, allowing for complex integration. The pyramidal cells of the cerebral cortex have about 40,000 contacts with other neurons. But the trade-off would be the chemical transmission involves a synaptic delay that makes the information travel much slower than it would if there was no synapse. So the two types of postsynaptic potentials produced by the neurotransmitters are the excitatory postsynaptic potential, EPSP, and the inhibitory postsynaptic potential, IPSP. And we alluded to both of those. And the voltage would change from negative resting membrane potential is going to go up toward the threshold. And then the excitatory postsynaptic potential is usually going to result from sodium flowing into the cell. Inhibitory postsynaptic potential is when the voltage becomes more negative than it is at rest. So an inhibitory postsynaptic potential can result from GABA. So GABA is going to cause, when GABA binds to postsynaptic terminals, they're going to open these channels, allowing for the influx of chloride. And you know, chloride is minus. It's not like sodium plus. Sodium plus causes excitation. Chloride minus is going to cause inhibition. So sodium influx with acetylcholine going to press the gas. It's going to speed up the neurotransmission. But if the chloride, because of GABA binding, GABA is unlike acetylcholine. So when GABA binds the postsynaptic terminal, it's going to open chloride channels, and the chloride channel is going to allow chloride to flow inwardly, causing a depolarization. So that's an excitatory postsynaptic potential. You see the membrane potential went up, and then that's the repolarization, and that is the returning back to the resting membrane potential. For inhibitory postsynaptic potential, you have a drop in the overall postsynaptic terminal. So that's a hyperpolarization. So there are different neurotransmitters that cause different types of postsynaptic potentials. There's a glutamate, and aspartate that produce excitatory postsynaptic potentials in the brain cells. And we said that GABA and glycine are both, upon binding to postsynaptic receptors, are going to elicit the influx of inhibitory electrolytes like chlorine. So a neurotransmitter might excite some cells and inhibit other cells, depending on the type of the receptors in the postsynaptic membrane. So acetylcholine and norepinephrine work in that way. So acetylcholine would excite a muscle cell, but inhibit a cardiac muscle cell due to the expression of different types of acetylcholine receptor on the different types of the muscle cells. So it is the receptor who gives the acetylcholine its identity. So if I give you $100 and I give somebody else $100, it's the same $100, but are you going to spend it the same way? That's exactly what happens with acetylcholine. So if acetylcholine binds to the cardiac cell, it's going to inhibit the cardiac cell. But if the same acetylcholine binds the skeletal muscle cell, it's going to cause a depolarization and a contraction. So even though both are muscle cells, both can contract, cardiac muscle cell can contract, skeletal muscle cell can contract, but acetylcholine is going to inhibit the cardiac cell from contracting, and acetylcholine is going to cause the skeletal muscle cell to contract. So that shows you the same acetylcholine has two different effects. The fire that boiled the egg make the egg harder, and the fire that cooked the egg, scrambled egg, make it soft. It's the same fire, but the way that it's used gives you a different effect. So acetylcholine is going to act differently. Now, what is the difference between summation, facilitation, and inhibition? Summation is the process of adding up postsynaptic potentials and responding to their net effect. That is the process of adding up postsynaptic potentials and responding to their net effects. And that occurs in the trigger zone. That occurs in the trigger zone. So some incoming nerve fibers may produce excitatory postsynaptic potentials, while others produce inhibitory postsynaptic potentials. So a neuron's response depends on whether the net input is excitatory or inhibitory. So the balance between excitatory postsynaptic potentials and inhibitory postsynaptic potentials enables the nervous system to make decisions. There are two ways the excitatory postsynaptic potential can be added to the threshold. There's temporal summation and there is spatial summation. In temporal summation, a single synapse would generate excitatory postsynaptic potentials that are so quickly that each is generated before the previous one fades. And that allows the excitatory postsynaptic potentials to add up over time to a threshold voltage that triggers an action potential. So we summate, we add up. It's like you're putting money in the bank and you keep accruing until you reach that threshold that enables you to buy what you want. So here, we cannot reach 55, minus 55. So we go up, minus 10, add to it another minus 5, add another minus 15, add another minus 20, until you hit minus 55. But for spatial summation, it is from several different synapses that add up to a threshold at the axon hillock. So here, you don't have one axon sending multiple adding up potentials. Here you have different. So now you have different people putting money in the bank. Let's all put, I put $5, you put 10, she puts 20, he puts 30, and then that is spatial. So you have different people, different neurons adding up to get us to the 55 threshold. But for temporal, it is one that just keeps on adding these different action potentials that would sum up to give me my long-awaited threshold to start my action potential. There we go. That's the temporal summation. In temporal summation, how many synapses do we have? One. How many neurons? One. See, they add. So there's an intense stimulation by one presynaptic neuron, and then you have excitatory postsynaptic potentials that spread from one synapse to the trigger zone. And these are the postsynaptic, and then the postsynaptic neurons would fire. Then you have spatial summation. In spatial summation, you would have simultaneous stimulation by several presynaptic. So now you see, you're not dealing with one neuron. You're dealing with you're dealing with 3 different neurons, but each one is adding up all of them. You see, the piggy bank is being filled by multiple different neurons, while here you have only one neuron. So that's spatial summation. That is simultaneous stimulation by several presynaptic neurons. So the excitatory postsynaptic potential is spread from several synapses to the trigger zone. This is the excitatory postsynaptic potentials that you see. This is an example of the summation. They sum up. That's why they call it summation. Whether temporal or spatial, we're adding up potential. You see, we're so low, minus 70. We're trying so bad to get to the threshold, minus 55. So we keep adding postsynaptic potential. Neurons can work in groups to modify each other's actions. The presynaptic facilitation occurs when one presynaptic neuron enhances another one. So neurons can work in groups to modify, and they cooperate with one another. So presynaptic facilitation occurs when one presynaptic neuron enhances another one, and that increases necessary synaptic transmission. An example would be facilitating the neuron, that is the cell F in the figure that we're going to see now, releases serotonin, which makes the voltage-gated calcium channels in the axon terminal in the figure. You see, there's no activity in facilitating the neuron, and these are the neurotransmitters.